Okay, so we're going to be starting our next lecture. This is the week 3 lecture for microbiology, general microbiology. This is for the nursing department. The focus for this week's lecture will be on microbial growth. All right, so let's get on with it. So a basic introduction for it. So we're going to be looking at the requirements for microbial growth. And microbial growth has both physical requirements as well as nutritional requirements. We're also going to be looking at the bacterial growth curve to see how the rate at which bacteria end up growing. And also we will be looking at some of the factors that affect bacterial growth. We'll also discuss how we count bacterial cells as well, or how we measure the quantity of bacteria. All right, so microbial growth refers to the process by which microorganisms such as bacteria increase in number or population. So growth is a sign that the microorganisms are actively metabolizing, which is necessary for their survival and their reproduction. In contrast to plants and animals, growth in microorganisms does not necessarily involve an increase in size or mass of individual organisms, but rather it's an increase in the total population of the microorganisms. Therefore, in the context of microorganism, growth typically refers to changes in population size rather than their individual size. Bacterial population growth primarily occurs through the process of binary fission. This means that a single bacterium can divide into two identical daughter cells that lead to an increase in the overall population. As long as the bacteria are not exposed to stress from nutrient depletion or environmental factors, they can continue to divide and propagate indefinitely. Because each division results in the doubling of population, bacterial growth can increase rapidly through geometric progression. So the requirements for microbial growth can be categorized into physical requirements and chemical requirements. The physical requirements include temperature, pH, and the osmotic pressure, while the chemical requirements include carbon, nitrogen, sulfur, and phosphorus, trace elements, oxygen, and other organic growth factors. For the physical requirements, again, we'll be looking at in detail temperature, pH, as well as osmotic pressure. Okay, temperature is a crucial factor that influences the growth, survival, multiplication, as well as the death of all living organisms. The rate of these processes is strongly influenced by temperature. For microorganisms, temperature plays an especially important role in their survival and growth. High temperatures can damage microbes by denaturing important proteins, such as enzymes and transport carriers, which are essential for their function. The microbial membrane can also be disturbed by temperature extremes. On the other hand, at very low temperatures, the microbial membrane can solidify and enzymes may not be able to function properly. So this can lead to a decrease in metabolic activity and growth rate. So in summary, temperature is one of the most important, if not the most important factor that affects microbial growth. And extremes in temperature can have damaging effects on an organism. So when we look at this slide, The minimum temperature is the lowest temperature that will support the growth of the microorganism, whereas the optimal temperature is optimum. This is the temperature, or again, more usually, the range of temperatures that support the fastest growth rate of microorganisms. Whereas the maximum temperature would be the highest temperature that will support the growth of microorganisms. When you get to areas above the maximum temperature, microorganisms usually end up being killed. Okay? And again, so high temperatures, we use this as we learned in the previous lecture by machines such as an autoclave, right, for sterilizing. The example we gave were the use of medical instruments. So if you look over here, when you have optimal temperature, you have a lot of growth. At the minimum temperature, there's minimal growth. And then when you come to if you come to temperatures that are high, maximum temperatures, again, what ends up happening is once you reach a higher temperature, then that growth, it declines sharply. Okay, so over here you're looking at, look at this temperature, this is how fast it grows. At this temperature, it's over here. So this temperature right here, whatever this number may be, is where bacteria will grow the fastest. Then if you come to this temperature over here, literally you're not going to get any growth. Bacteria will start to die at this point. All right, so we're going to be looking at specific classifications of bacteria based on temperature, so such as psychrophiles, mesophiles, and thermophiles. All right, there are two groups of microorganisms that can grow in cold temperatures, often referred to as cold-loving. The first group is the true microphiles, which are sensitive to temperatures over 20 degrees centigrade. They have an optimum growth temperature of about 15 degrees centigrade or less. These microorganisms are typically found in very cold environments, such as the North Pole or at ocean depths. Seldom do they cause disease or food spoilage. Their temperature range, again, is between minus 10 centigrade to positive 20 centigrade. Now, the second group are the psychotrophs, which have an optimum growth temperature of about 20 to 30 degrees centigrade. These are responsible for most low temperature food spoilage. These microorganisms can grow in a wider temperature range from 0 degrees centigrade to positive 30 degrees centigrade. Now, unlike true psychrophiles, psychotrophs are not excessively found in extremely cold environments. Mesophiles are microorganisms that grow best at moderate temperatures that range from 20 to 45 degrees centigrade. Commonly, they're referred to as middle-loving organisms. Now, most bacteria, including many pathogens and common spoilage organisms, are mesophiles. The optimum growth temperature for mesophiles is about typically around 37 degrees centigrade. Many mesophiles have adapted to live in the bodies of animals, including humans. So some examples of mesophilic bacteria include E. coli and Streptococcus pneumoniae. Because mesophiles are so common, they play a significant role in many of the different biological processes, including disease and food spoilage. Heat-loving microorganisms, or thermophiles, are microorganisms that have an optimum growth temperature between 50 to 60 degrees centigrade. Many thermophiles can't grow below 45 degrees centigrade, and we find them in sunlit soil as well as hot springs. Now, some of these thermophiles, they have the ability to form extremely heat-resistant endospores, which allows them to survive in harsh environments. Now, with that said, there are also extreme thermophiles, also known as hyperthermophiles, which have an optimum growth temperature of around 80 degrees centigrade or higher. Now, most extreme thermophiles are archaeobacteria, and they are commonly found in volcanic and ocean vents. These organisms have adapted to survive in some of the most extreme environments on the Earth, including deep sea hypothermal vents and volcanic areas. So this slide here is just showing you the temperature range for the psychrophiles, psychotrophs, the mesophiles, thermophiles, as well as the hyperthermophiles. So if you look over here for the hyperthermophiles, again, the temperature range that they prefer from 65 all the way to over 110, according to this graph that they have over here. So again, notice that there's a very wide range for the different types of bacteria who thrive in different temperature conditions. So again, as you can see, temperature, it's a very important detriment in terms of bacterial growth. All right, so next is pH. pH refers to the negative logarithm of hydrogen ion concentration. And this is an important factor that affects microbial growth. Microorganisms have adapted to grow within a specific pH range, and drastic variations in the cytoplasmic pH can disrupt the plasma membrane, or it can even inhibit the activity of enzymes and membrane transport proteins. This is the same for humans as well, which is why our blood has to stay within a specific, very narrow pH as well, 7.35 to 7.45. Right? You go below or above, we get very sick, we can die. So again, this is very important also. Same thing with temperature, as we discussed before. If our body temperature drops below or was above a certain point, we get very sick. So again, bacteria, same thing. They have not only specific temperatures, but now also we're looking at specific pH that they need to stay within. So the optimal pH range for growth will vary depending on the microorganism. Some of these organisms, they grow best at a neutral pH of about 7. Others, they prefer acidic or alkaline conditions. So for example, acidophiles are microorganisms that grow best at a pH of about 5.5, while alkiphiles, they grow best at pH levels above 8.5. Now, most bacteria prefer a neutral pH range, typically between 6.5 to 7.5. Molds and yeast can tolerate a much wider pH range, but they prefer a slightly acidic environment. The optimal pH range for them would be between 5 and 6. Now, acidity inhibits most microbial growth, and this property is utilized in food preservation techniques like pickling. So alkalinity also inhibits microbial growth, but it's not commonly used for food preservation. So the acidic products of bacterial metabolism can interfere with the growth of microorganisms, making the use of buffers essential to stabilize pH levels. Buffers are compounds that can also that have the ability to absorb or release hydrogen ions, and this helps to maintain a stable pH. So proper pH control is critical in many microbiological applications, including food preservation, as well as industrial fermentation, and also in medical diagnostics. So now let's take a closer look at acidophiles, neutrophiles, and alkophiles. Acidophiles, when you look at this word, means acid-loving, and again, they grow at very low pH, okay? Hence, acid-loving, acidophile. So their optimum pH is between 0 to 5.5 and less. So lactobacillus produces lactic acid. So again, it's able to tolerate mild acidity. Right, neutrophiles grow best between pH of 5.5 to 8.0. They include most human pathogens such as E. coli as well as Pseudomonas aeruginosa. Alkaphiles are also known as alkali-loving microorganisms, and they're a type of bacteria that thrive in alkaline or high pH conditions. They have an average pH range that they like of between 7.5 to 14. Examples of alkaphiles include Vibrio chlorei, which has an optimal pH of about 9, and there's also Agrobacterium, which is a soil bacterium that grows best at a pH of about 12. So we can summarize by saying that alkaphiles are unique in their ability to survive and thrive in extremely alkaline environments such as cemented soils and alkaline springs, even soda lakes. And we can further say that these microorganisms have adapted to their extreme environments by developing specialized enzymes and transporters that can function at these high pH levels. Now, osmotic pressure refers to the minimum pressure that's needed to prevent the inward flow of water across a semi-permeable membrane. In microbial growth, the osmotic pressure of the surrounding environment can have a significant impact on the survival and growth of microorganisms. All right, there are 3 types of solutions based on their osmotic pressure. There's hypotonic solution. Let's start off with this one. Hypotonic solution is a solution with a lower solute concentration than the surrounding environment. Water flows into the cell, causing it to swell and potentially burst. Next, let's go to isotonic solutions. Isotonic solution is a solution with the same solute concentration as the surrounding environment. So there's no net movement of water into or out of the cell. Now, a hypertonic solution is a solution with a higher solute concentration than the surrounding environment. Water flows out of the cell, causing it to shrink and potentially die. Microorganisms have various mechanisms that helps them cope with changes in osmotic pressure, such as the synthesis of compatible solutes, which help to balance the concentration of solutes inside the cell and the external environment. So when we're looking over here, what we see is that this, first of all, is an isotonic environment, an isotonic solution. So the amount of water that's coming into the cell and the amount of water that's going out to the cell, there's no change, right? So nothing happens to the cell. It's equal, okay? The solute concentration that's within the water is the same as the solute concentration that's inside. So what happens? There's no net movement. Everything's the same. Now, what do we have? We have a hypotonic solution, right? What does that mean? That means the solute concentration inside the cell is greater than what is outside the cell. So what's going to happen? The cell is going to try to take in much more water to be able to recruit, to take in these solutes inside. So therefore, what's going to happen? The cell it may lyse, right? It may burst, okay? It'll get big because you're getting too much water coming inside. Then we have a hypertonic solution. Now in a hypertonic solution, what's happening is that, again, with the environment, it's hypertonic. There's too much solutes than what the cell would like. Okay, there's much less solutes inside the cell and tons and lots of solutes outside. So the amount of water that goes in carries so much solute that the cell is trying to push out as much water as it can. Therefore, the cell starts to shrivel, right? And it will shrink. And the term that we use for that is that it can shrivel up and die, or plasmolysis. So again, to go into more details, let's look at the osmotic pressure now. So hypertonic environments with high salt or sugar levels can cause plasmolysis in bacteria, where the cytoplasm shrinks away from the cell. Some bacteria are adapted to high salt environments. And again, these are known as extreme or obligate halophiles. And they require high osmotic pressures to survive. Other bacteria known as facultative halophiles can tolerate high osmotic pressures, but they do not require it for survival. So halophiles are microorganisms that require moderate to large salt concentrations for growth. We find them in various habitats such as oceans and salt lakes. Extreme or obligate halophiles require very high salt concentrations, while facultative halophiles can grow in the absence or presence of high salt concentrations. Now, obligate halophiles are found in habitats such as the Dead Sea, as well as brine vats. Facultative halophiles, they don't require high salt concentration for growth, but they can tolerate 2% salt or more. The environment of every organism must contain all the necessary substances for energy generation and cellular biosynthesis. The substances that are utilized for bacterial growth are referred to as nutrients or nutritional requirements. Now, in the laboratory, bacteria are grown in culture media that provide all the essential nutrients in a solution for bacterial growth. Carbon is a crucial element for the structure and function of all living cells, making up about 50% of their dry weight. Bacteria have different nutritional requirements for carbon, which chemoheterotrophs obtain from organic sources such as lipids, proteins, and carbohydrates, while chemoautotrophs and photoautotrophs, they will obtain it from carbon dioxide. Okay? Chemoheterotrophs use their energy source to obtain carbon, while chemoautotrophs and photoautotrophs use carbon dioxide as their sole carbon source. Nitrogen is a vital element for the formation of amino acids, DNA, and RNA in bacterial cell, making up about 14% of dry cell weight. Bacteria can obtain nitrogen from different sources, such as proteins, ammonium, nitrogen gas from the atmosphere, and nitrates that dissociate in salts. Most bacteria use proteins as a source of nitrogen, while some bacteria live free in soil or associated with legumes to fix atmospheric nitrogen. These bacteria are known as nitrogen-fixing bacteria, and they're essential in agriculture as they can improve soil fertility and crop yield. Sulfur is an essential element that's used to form proteins and some vitamins, while phosphorus is used to form DNA, RNA, ATP, and phospholipids. Both elements are commonly found in protein and are important for bacterial growth. Sulfur can also be sourced from hydrogen sulfide and sulfite salts, while phosphorus is mainly sourced from inorganic phosphate salts and buffers. Oxygen is an essential component for many organisms for generating energy from nutrients. Microorganisms can be classified based on their oxygen requirements. Obligate aerobes require oxygen for growth, whereas obligate anaerobes can only grow in the absence of oxygen. Facultative anaerobes can grow in both the presence and absence of oxygen, while aerotolerant anaerobes do not use oxygen but can tolerate its presence. Microaerophiles require a low level of oxygen for growth. Without having to go into details about what we just explained in the previous slide, examples of obligate aerobes include Pseudomonas, and these are common in nosocomial pathogens. Example of facultative anaerobe would be Staphylococcus, E. coli, yeast, and many other intestinal bacteria. For obligate anaerobes, example would be Clostridium bacteria that causes tetanus and botulism. And For aerotolerant anaerobes, an example would be lactobacillus, which carries out fermentation regardless of the oxygen presence. And microaerophiles, example would be Campylobacter. Organisms require certain essential organic compounds for growth as they're unable to synthesize them themselves, and they must obtain them through the environment. These essential compounds are known as growth factors and divided into three categories. We have the purines and pyrimidines, the amino acids, and vitamins. Purines and pyrimidines are required for the synthesis of nucleic acids such as DNA and RNA. Amino acids are needed for the synthesis of proteins, and vitamins are required as coenzymes and functional groups of certain enzymes. Some bacteria lack the enzymes required for the synthesis of certain vitamins, so they must obtain them directly from the environment. Examples of growth factors include amino acids and purines and pyrimidines. A culture medium is a nutrient-rich material that's used to promote the growth of microorganisms in a lab setting. The medium must be sterile and contain appropriate nutrients that will support the growth of the specific microorganism that's being studied. It must also be incubated at the appropriate temperature, as well as the environment, to promote optimal growth. Now, the process of growing and multiplying microbes in or on a culture medium is called culturing. Solid media are nutrient materials that contain a solidifying agent such as agar, which was first used by Robert Koch. Agar is a unique solidifier as it melts above 95 degrees centigrade, and it doesn't solidify until it reaches about 40 degrees centigrade. Additionally, agar cannot be degraded by most bacteria. It's made from a polysaccharide that's produced by red algae. Initially, it was used as a food thickener, but now it's widely used in microbiology as a solidifying agent for culture media. Chemically defined media is a nutrient material with a known chemical composition. It's typically used for chemoheterotrophs and must contain an organic source of carbon and energy. It may also include amino acids, vitamins, and other essential building blocks that are required by the microbe. However, it's not commonly used due to its high cost. It doesn't come cheap. Right, complex media is a nutrient material commonly used for the growth of heterotrophic bacteria and fungi. The exact chemical composition of complex media is unknown as it's made up of extracts from yeast, meat plants, and protein digests. Now, energy, carbon, nitrogen, and sulfur requirements are primarily met by protein fragments, which we call peptones, while vitamin and organic factors are provided by meat and yeast extracts. Complex media is available in two forms: nutrient broth, which is a liquid media, and nutrient agar, which is a solid medium. The composition of the complex media, it could vary slightly from one batch to the next. Anaerobic growth media are used to cultivate microorganisms that cannot tolerate oxygen. Reducing media are the most commonly used type of anaerobic growth media. These media contain ingredients that chemically bind with oxygen and eliminate it from the medium. In special culture techniques, certain bacteria require a unique growth conditions. Capnophiles are bacteria that require high levels of carbon dioxide and low levels of oxygen to grow. These conditions are similar to environments found in the respiratory and intestinal tracts and other tissues. Capnophiles are grown using specialized incubators that provide controlled carbon dioxide levels. These incubators can also be used to grow microaerophiles, which require low levels of oxygen and higher levels of carbon dioxide for optimal growth. All right, here's an anaerobic jar. All right, so again, notice what we have over here is this is a chamber, and this chamber essentially will remove oxygen and provide higher levels of carbon dioxide. Okay, that's essentially it, right? So you have this envelope over here that contains sodium bicarbonate and sodium borohydride, which is releasing carbon dioxide and hydrogen gases. So again, then you have a test strip that will change colors to show that the proper conditions are met. Right. So this is ensuring that yes, we have met a specific level of concentration of these gases that again that the bacteria would favor, which would be found in these plates over here, these petri plates, or petri dishes as they're also called. As I said, sometimes you have a chamber. Okay, this is an airtight chamber, and you can do your streaking, whatever you need to do over here. The introduction of microbes, you will do it in this airtight chamber. This ensures that no oxygen is inside over here when making transfers. And again, so this is this anaerobic chamber as it's called. Selective media are used to isolate specific microorganisms from mixed cultures by suppressing the growth of unwanted bacteria and promoting the growth of the desired ones. Some examples of selective media include Cyberrod's dextrose agar, which has a pH of about 5.6, and this discourages bacterial growth. And we use this to isolate fungi. Brilliant green agar uses green light to selectively inhibit gram-positive bacteria, and it's used to isolate gram-negative salmonella. Salmonella typhi. All right, so bismuth sulfite agar is used to isolate Salmonella typhi by inhibiting the growth of most other bacteria. So very simplistically, selective media will encourage the growth of desired microbes while suppressing the growth of unwanted microbes. Differential media is a type of culture medium that helps in differentiating between colonies of different microbes. One example of this type of media is blood agar. Blood agar is a differential media that's used to distinguish bacteria based on their ability to destroy red blood cells, a process that's known as hemolysis. Hemolysis is observed as a clearing or discoloration of the media around the bacterial colony. So, for instance, Streptococcus pyogenes, which is a pathogenic bacteria, shows beta hemolysis on blood agar, whereas other Streptococcus species displays alpha hemolysis or gamma hemolysis. Mannitol salt agar is a type of culture medium that's both selective and differential. It's used to select for and differentiate Staphylococcus aureus from other organisms. The medium contains a high concentration of salt, about 7.5% sodium chloride, which inhibits the growth of most other organisms. It also contains mannitol, which is fermented by Staphylococcus aureus to produce acid. This fermentation causes a pH indicator in the medium to change color, allowing for easy identification of Staphylococcus aureus colonies. Mac-Konicke agar is a type of selective and differential media used to isolate and differentiate gram-negative bacteria, especially Salmonella. The selective properties are due to the presence of bile salts and crystal violet, which inhibit the growth of gram-positive bacteria. The differential properties are due to the inclusion of lactose and pH indicator in the medium. Lactose fermenting bacteria produce pink or red colonies due to the acid production, while non-fermenting bacteria remain colorless. The use of this media is particularly useful in clinical microbiology to isolate enteric bacteria such as Salmonella from clinical samples. Enrichment culture is a technique used in microbiology to encourage the growth of microorganisms that may be present in very small numbers. Unlike selective medium, which is designed to suppress the growth of other microbes, enrichment culture does not necessarily have this effect. This method is particularly useful for analyzing fecal and soil samples. So, after incubating a sample in an enrichment medium, the greater number of microorganisms that are present increase the likelihood of positive identification of the microbe that's in question. All right, in microbiology, a pure culture refers to a culture that contains only one species or strain of microorganism. A colony, also known as a colony forming unit or CFU, is a population of cells that originates from a single cell or spore or a group of attached cells. Most clinical and environmental specimens typically contain multiple microorganisms, so isolating individual microorganisms is necessary in order to obtain a pure culture. The most commonly used method for isolation is the streak plate method, which involves spreading a sample onto a solid growth medium and dragging a sterile loop across the surface to dilute the cells and separate them into individual colonies. The streak plate method enables the isolation of pure cultures, which are essential for conducting further research and experimentation in microbiology. So over here, you can see this streak plate method. So what you do is, again, you take an inoculating loop and you first smear it like this. Then you'll rotate the petri dish and then you will smear it like that. Then you will continue to rotate and then streak it as such. And you will then see the resulting colonies being isolated. Preserving bacterial culture is an important aspect of microbiology, and two common methods for long-term storage are deep freezing and lyophilization. Deep freezing involves storing bacterial cultures at very low temperatures, typically between -50 to -95 degrees centigrade. And what this does is that it slows down the metabolism of the bacteria and helps to prevent degradation. Another method is lyophilization. Another term for this is freeze drying, and this involves freezing the bacterial culture at temperatures between -54 to -72 degrees centigrade, and then dehydrating it in a vacuum to remove any remaining water. And what this will do is that it will prevent the growth of microorganisms and ensures that the bacteria culture can be stored for an extended period of time without losing viability. Both these methods are commonly used in microbiology labs for preserving bacterial cultures. Bacterial cell is the process by which a bacterial cell replicates itself, and it occurs mainly through binary fission. Binary fission is the process where the parent cell divides into two identical daughter cells. However, a few bacterial species reproduce through budding. This is where a small bud grows out of the parent cell and eventually detaches to form a new cell. Now, the generation time is the time that's required for bacterial cells to divide and for its population to double. Generation time can vary significantly among bacterial species. Some bacteria are able to divide every 20 minutes, such as E. coli, while others require over 24 hours to divide. We can say that the generation time is influenced by a variety of factors, such as nutrient availability, temperature, and pH level. If we understand these factors, it allows us to better study bacterial growth and reproduction. The growth of bacteria in a batch culture can be divided into 4 different phases. Collectively, we call this the growth curve. The first phase is the lag phase, where bacteria adjust to the new environment and prepare for rapid growth. The second phase, which is called the log phase or exponential phase, this is where bacteria experience rapid growth and division. This happens because there's sufficient nutrients and space for them to reproduce. Now, the third phase, the stationary phase, is where bacterial growth slows down and reaches a plateau. And this is due to the depletion of nutrients or the accumulation of waste products. The final phase is the death phase. This is where the number of viable bacteria decrease due to the exhaustion of nutrients and the accumulation of waste products. The lag phase is the initial phase of bacterial growth when cells are introduced to new environmental conditions. During this phase, bacteria undergo a period of adjustment and exhibit little or no cell division. This can last anywhere from an hour to several days, depending on the bacterial species as well as the environmental conditions. While cells are not dormant during the lag phase, they're not actively dividing either. Instead, individual organisms grow in size as well as they undergo a period of intense metabolic activity, including DNA and enzyme synthesis. And this is in preparation for the log phase of growth. So, again, even though it appears like nothing's happening within the cells, there's activity there. Now, the log phase is a period of bacterial growth where the number of cells produced is greater than the number of cells dying. So this phase is also known as the exponential growth phase, since the bacterial population doubles at regular intervals. So during this phase, cells are at their highest metabolic activity and cellular respiration is most active. However, cells are also the most susceptible to adverse environmental factors at this stage, such as radiation and antibiotics. So the log phase is a critical period for the study of bacterial growth, as this is when the bacterial populations are actively dividing and are most vulnerable to external factors. The stationary phase, this is a period of bacterial growth when the number of cells produced equals the number of cells that are dying, and this results in a period of equilibrium. So during this phase, the overall cell number doesn't increase. The metabolic activity of the surviving cells, it slows down, and then this is what helps stabilize the population. Now, several factors can slow down microbial growth during the stationary phase. This includes the accumulation of toxic waste materials, acidic pH of the media, the limited nutrients that would be available, as well as insufficient oxygen supply. So the stationary phase is an essential phase in bacterial growth, as it represents a period of stability where bacteria can adapt to the environment and resist unfavorable conditions. The death or decline phase is the period of bacterial growth when the population size begins to decrease. During this phase, the number of cells dying is greater than the number of cells that are produced, and this results in a logarithmic rate of decline. Cells lose their ability to divide during this phase, which is also known as the logarithmic decline phase. Okay. Some populations may die out completely, while a few cells may remain alive for a long period of time. Okay, so over here we have the y-axis, which shows the number of bacteria, and then we have the x-axis, which shows time. So number one, over here, this is the log phase. You can see there's nothing going on. Everything's pretty much stationary. Okay, number of cells are not being produced too much, and the number of cells that are dying, more or less, it's the same. All right. So again, we have a flat line that's going across on our next curve, on the x-axis. Okay. Now we come to the next phase, the log phase, number 2. And now what we see is the number of bacteria increasing exponentially. Okay, so again, literally it continues to double and double and double and double and double over a certain period of time until it reaches a plateau over here. And at this peak over here, when it reaches this peak, then it plateaus. Then again, it flattens out again for a certain amount of time. So let's assume that this lag phase is maybe an hour. If you look over here, maybe this is an hour and a half, perhaps. Okay, this is right about the 5th hour mark. So during the stationary phase, remember the number of cells that are being produced and the number of cells that are dying, they're nearly the same, right? Which is why, again, we have this flat line that's going across, this plateau that takes place. Then we have the last phase, the death phase. In death phase, the number of cells that are dying are far greater than the number of cells that are being produced. So again, now we get this decline. Okay, so measuring microbial growth is an important aspect of microbiology, which involves determining the number and size of microorganisms in a sample. And there's several methods for measuring microbial growth. And we can divide these into two broad categories: direct method and indirect method. The direct method involves physically counting the microorganisms or measuring their size. Plate counts, filtration, most probable number, and direct microscope count are examples of direct methods. The plate count involves spreading a sample of microorganisms on a growth medium and counting the number of colonies that grow. Filtration involves filtering a sample of the microorganisms through a membrane and counting the number of colonies that grow on the membrane. The most probable number involves diluting a sample of microorganisms and determining the probability of growth based on the number of positive and negative tests. Direct microscopic count involves counting the microorganisms directly under a microscope, whereas indirect method involves measuring a physical or chemical property of the microorganism that correlates with their growth. Turbidity, metabolic activity, and dry weight are examples of indirect methods. Turbidity involves the cloudiness of the liquid culture, which increases with the number of microorganisms present. Metabolic activity involves measuring the production of a specific metabolite, a specific metabolic product, such as carbon dioxide or acid, which indicates the presence of living microorganisms. Dry weight involves measuring the weight of a sample of microorganisms after it has been dried, which correlates with the number of microorganisms present. Plate counts are a common direct method for measuring microbial growth, which involves spreading a sample of microorganisms onto a growth medium and counting the number of colonies that grow. So the advantage is that it measures viable cells. However, it's slow. It could take 24 hours or longer for visible colonies to start to appear. Other thing is that, again, you're only seeing counts between 25 to 250 colonies where we get an accurate measure. And you have to perform serial dilutions to get the appropriate numbers or plates. So if you look over here again, what you're doing is, in this slide, we have the original inoculum. So this is the original tube. And then again, what's the dilution at? 1:10 ratio. So you take that and then you're going to plate it. Then from there, you're going to take a sample and introduce it over here. So you're going from 1:10 to, now you're diluting it. Now this is a 1:100. So now you'll take this and then you'll make a sample over here. You'll plate it here. Then you'll take from this dilution, you'll put into another, you'll dilute it even more. So now you have what? 1:1000. And then from here, you go to 1:10,000. And then from 1:10,000, you go from 1:100,000. So each time, again, what you're doing is you're diluting it. Again, the issue is that you have to do many of these, and also all of this is taking lots of time. Okay. Now, one type of plate count is the pour plate method. This involves introducing a small amount of the inoculum into an empty petri dish and adding liquid nutrient medium that's been kept at a specific temperature. Then you take that mixture and it gets mixed gently, and then it's allowed to get solidified, and then it gets incubated to allow for the growth of the microorganisms. Now, with that said, there's some disadvantages to the pour plate method. First, it's not useful for heat-sensitive microorganisms since they may be killed by the high temperatures that are required for the nutrient medium. The next thing is that the colonies that grow, they may appear under the agar surface, which can be a little bit difficult to count accurately. The spread plate method is another type of plate count that involves introducing a small amount of the inoculum, usually about 0.1 to about 0.5 ml, onto the surface of a petri dish and then spreading evenly across the agar surface with a sterile glass rod. Now, this method has the advantage of producing colonies that are on the surface of the agar, making it easier to count and distinguish from other colonies. Additionally, since the colonies are not exposed to melted agar, they may be less likely to be damaged or altered during the growth process. So when you look over here for the pour plate method, so what do we have? We're taking this bacterial dilution and we first inoculate an empty petri dish or an empty plate, as they say. After that, then the addition of the melted nutrient agar is added. Once that's been added, then you swirl it around to mix it, and then you wait. Okay, the colonies grow, and then it gets solidified into the medium. Right? For the spread plate method, again, you take the bacterial dilution and inoculate the plate onto the solid medium. Okay, so now the medium is already here. Next, you take the sterile glass rod and then you spread it. Then you wait for the colonies to grow. All right, filtration is a direct method for measuring microbial growth that's often used to measure small quantities of bacteria in environmental samples, such as fecal bacteria in lake or ocean water. Now, to perform filtration, a large sample of the water, typically about 100 ml or more, is filtered through a membrane that retains the bacteria. The filter is then transferred onto a petri dish containing a nutrient medium and then ends up being inoculated to allow the bacteria to grow. Now, the colonies that grow on the filter can then be counted to determine the number of bacteria that are present in the original sample. So filtration has several advantages as a method for measuring microbial growth, which include its ability to concentrate bacteria from large volumes of water and its ability to remove interfering substances that may be present in the sample. However, it can also be time-consuming and may require specialized equipment to perform. The most probable number, MPN, is a direct method for measuring microbial growth that is commonly used to measure bacteria that will not grow on solid media. Now, to perform the MPN assay, a sample is diluted repeatedly and inoculated into several broth tubes for each dilution point. The tubes are then observed for growth, and the number of positive tubes at each dilution point is recorded. Now, this data is then analyzed using statistical methods to determine the most probable number of bacteria present in the original sample, with a 95% probability that the bacterial population falls within a certain range. Now, the MPN method has several advantages, including its ability to measure bacteria that do not grow on solid media, and its ability to provide an estimate of the number of bacteria present in a sample. However, it can also be time-consuming and may require specialized equipment to perform. All right, the direct microscopic count is a direct method for measuring microbial growth that involves placing a specific volume of a bacterial suspension, usually about 0.1 ml, on a microscope slide with a special grid. A stain is then added to visualize the bacteria, and the cells are counted and multiplied by a factor to obtain the concentration. Now, the direct microscopic count method has several advantages, including its ability to provide rapid results without the need for incubation time. However, it also has several disadvantages, including its inability to always distinguish between live and dead bacteria, as well as the difficulty in counting motile bacteria, and the requirement for a high concentration of bacteria to achieve accurate results. All right, so over here you can see that we have a slide that has a special grid on it. You take your sample, you put it on, and then you observe it, and you're able to count. So again, very fast way to take a count, the sample. Turbidity is an indirect method for measuring microbial growth that's based on the fact that as bacteria multiply in a liquid media, it becomes turbid. To measure turbidity, a spectrophotometer is used to determine the percentage of transmission or absorbance, which is then multiplied by a factor to determine the concentration of bacteria. Now, the turbidity method has several advantages, including its ability to provide rapid results without the need for incubation time. Now, with that said, it also has several disadvantages, including its inability to distinguish between live and dead bacteria, as well as the requirement for a high concentration of bacteria, which is typically about 10 to 100 million cells per ml, in order to achieve accurate results. So if you look over here, what you're looking at is this. So here's a light source, here's a test tube, light is exposed, and this instrument over here is measuring how much light is able to pass through. So the more light that passes through, that means the less bacteria are present. Okay, so if you look over here in this tube, there's lots of bacteria. So again, as light is passed through over here, a lot of that light ends up, because this is concentrated with lots of bacteria, very little light is able to pass through and to be measured by this device over here. So most of the light gets bounced back off. Little light pass through, or very little light gets absorbed in this instrument here. So this solution is turbid. Okay, there's lots of particles inside that are floating, blocking the obstruction of the light to pass through. Now, metabolic activity is an indirect method for measuring microbial growth that's based on the fact that as bacteria multiply in a liquid media, they produce certain products such as carbon dioxide and acids. The metabolic products can be measured using extensive methods to indirectly determine the bacterial population. Dry weight is another indirect method for measuring microbial growth that involves centrifuging bacteria or fungi in a liquid media and then weighing the resulting cell pellets. This method doesn't distinguish live and dead cells, so it doesn't provide a complete picture of microbial growth. All right, so that's all for this lecture. Our next lesson, the topic is supposed to be genetics, microbial genetics. Okay, so thank you for watching this lecture. Please be sure to take good notes. If you haven't taken notes, please review this lecture again and take good notes, and be sure you're studying well. Thank you so much, and we'll see you guys next time.